Staining cells is based on the ability of certain structures to differentially bind or absorb the stain while the rest of the cell does not. Hi, welcome to the mini class, and today we're going to talk about how we stain cells. When we look at natural cells, actually the contrast is pretty low and we don't really see structures or any detail. So I want to show you how the principle of staining cells actually works. So, for example, let's take a pen and draw a cell. And you can see that I'm drawing the cell and its nucleus and maybe some ER and some mitochondria. As you can see, there's very little contrast and we don't see many details at all. However, if we add some stain, in this case powder, to the surface and to the total surface of the cell, as you can see here, to the drawing, we're not staining something specific, but rather adding color, in this case powder, to everything. Here we go. And when we remove that powder, we see a picture with contrast. This is how staining works in the cell. And it is through these processes of specific staining, different organelles in our cells, we can see contrast and see more detail. In the demo, the powder was able to stick to the moist ink, while the surrounding sheet of plastic did not. If we want to stain the nucleus, for example, we might want to use a chemical or a reagent that specifically binds DNA. And if this stain has a fluorescent blue color, then all the nuclei of the cells will stain fluorescently blue. Thus, for each organelle, scientists have developed colored dyes that selectively bind to that organelle and stain it, revealing its shape, size, and position within the cell. I've asked Professor Marcelo Ehrlich to show us some cells under his microscope.
My name is Marcelo Ehrlich, and this is an immunofluorescence microscope. Today, we are going to use this microscope to observe live HeLa cells, which are human cervical carcinoma cells, at very high resolution. In order to observe specific intracellular compartments and organelles, we have stained these cells with colors so as to be able to observe the nuclei and mitochondria. This Petri dish that I now hold has 200,000 of these living HeLa cells. Let's go to the microscope and look at them.
My name is Antoni van Leeuwenhoek and this is my microscope.
It's totally amazing how microscopes have developed. Let's look at some of the cells Marcelo has prepared for us. Without staining, it's hard to pick out details in these HeLa cells, and you might even have trouble making out where one cell begins and another ends. Staining the nuclei blue immediately helps us identify individual cells. Surprisingly, the mitochondria, stained here in red, are far more abundant than what we might expect. Cells have loads of mitochondria dispersed throughout the cytoplasm. The Golgi apparatus, on the other hand, tends to be more localized, as is seen here in green. Here in this micrograph, we see the boundaries of each cell by staining the outer cell membrane. Each cell contains an extensive network of intracellular membranes, the endoplasmic reticulum, stained in green. We can confirm the ghost in the center of the cell is the nucleus, the organelle that holds our DNA, the stuff genes are made of. In the following lesson, we will further explore just how DNA works and how information stored in DNA is translated to make proteins that build our bodies.